This method can help answer key questions in the field of gene expression regulation, such as how RNA binding proteins assemble on particular RNAs in vivo and thereby impose post-transcriptional gene regulation.
The main advantage of this technique is that it does not need any cloning or genetic manipulation. It can be adapted to any model organism or cell type.
To design oligonucleotides, analyze the secondary structure of the mRNA or a fragment thereof using online tools. First, enter the nucleotide sequence in the empty box. Then in the basic options box, select minimum free energy and avoid isolated base pairs. In the output options box, select interactive RNA secondary structure plot and finally click the proceed button. A new window will pop up that displays the secondary structure of the mRNA of interest. Select at least 3 different 21 to 24 nucleotide long sequences within the mRNA of interest, preferentially in regions lacking extensive secondary structures and located in 3' untranslated regions. Select regions with a guanidine-cytosine ratio close to 50% and lacking nucleotide tandem repeats to avoid potential formation of hairpins or self-annealing. Manually design 2' methoxy-modified RNA oligonucleotides bearing a biotin moiety at the 3' that are fully complementary to the selected regions within the desired target mRNA. Use a suitable online tool to adjust the melting temperature of the RNA hybrids to between 60 and 65 degrees and having a good linguistic sequence complexity.